Hello everyone, I am Nivedita Jena from Ambika Prasad Research Foundation. Welcome to all of you to our channel Biodiversity and Conservation. So let's discuss about today the bacterial characterization. Before going to brief characterization methods of microbes, first we have to know that what are microbes and what they define. The microbes are the organisms that are too small to be seen without using a microscope. So they include things like bacteria, archaea, and single-cell eukaryotes, cells that have a nucleus, like an amoeba or a paramecium. Sometimes we call viruses microbes too. Uh, the term is very general. It is used to describe many different types of life forms with dramatically different sizes and characteristics like bacteria. And microbes live in water, soil, and in the air. The human body is home to millions of these microbes too, also called microorganisms. Types of microbes. As microorganisms are one of the most diverse organisms and they include bacteria, fungi, archaea, protozoa, algae, and viruses. So the first one is bacteria, which are the simplest of creatures that are considered alive. And bacteria are everywhere. They are in the bread you eat, the soil that plants grow in, and even inside of you. They are types of microbes, as the microorganisms are one of the most diverse organisms, and they include bacteria, fungi, viruses, algae, protozoa, and archaea. The first one is bacteria, which are the simplest of creatures that are considered alive, and bacteria are everywhere. They are in the bread you eat, and the soil that plants grow in, and even inside of our body. They are very simple cells that fall under the heading prokaryotic. That word means they do not have an organized nucleus. And bacteria are small single cells whose whole purpose in life is to replicate. And then coming to the fungi, fungi are classified within their own kingdom. The kingdom fungi, while some are in the kingdom protista. A fungus is neither a plant nor an animal. It is similar to a plant, but it has no chlorophyll and cannot make its own food like a plant can through photosynthesis. They get their food by absorbing nutrients from their surroundings. Many fungi play a crucial role in decomposition and returning nutrients to the soil. They are also used in medicine. An example is the antibiotic penicillin, and as well as in industry and food preparation. The aim of bacterial characterization is to obtain a complete collection of data describing the properties of a prokaryote pure culture, that is, to form a description. The aim of identification is to equate the properties of a pure culture with those of a well-characterized taxon, and to confirm identity, establish relationship between isolates, and understand the behavior, phenotypic behavior, and pathogen detection, and correct diagnosis and treatment. The bacterial characterization is important. Before entering to the characterization part, we have to first look out the methods of bacterial identification. Then we can briefly understand the characterization methods of microbes like bacteria. First one is the genotypic method. Genotypic microbial identification methods are more reliable than phenotypic methods because nucleic acid sequences are conserved in most microbial species, and genotypic methods for microbial identification include DNA-DNA hybridization, PCR, 16S and 23S rRNA sequencing, multilocus sequence typing, virus sequencing, DNA probes, and analytical ribotyping. And these methods are technically challenging and require more expensive analytical equipment. For these regions, Genotypic identification methods are limited to critical microbiological investigations such as product failure investigations. Then coming to the proteotypic method. So this method is used to express a protein spectrum that assists in identification of microorganisms. This method basically uses the chemical and physical mode to bring out emissions from the microorganism. And then coming to the phenotypic method. So the phenotypic method is used to metabolic differences between different species to identify microorganisms. And generally, they involve a combination of gram staining, culture, and biochemical tests. So these are some examples of bacterial identification method. The phenotypic methods include biochemical assays based on physiological reactions and immunological methods and also fatty acid profiles. Then prototypic methods include Fourier transform, infrared spectroscopy, which is also said FTIR spectroscopy, and matrix-assisted laser dispersion ionization, time-of-flight mass spectrometry, which is also commonly known as multi-tough mass spectrometry. And the last one, genotypic methods include the nucleic acid amplification techniques and genetic fingerprinting or ribotyping. Then there are some factors on which bacterial identification method depends. They include chemical characteristics, morphological characteristics, ecological characteristics, genetic and antigenic characteristics, and pathogenicity, cultural and metabolic characteristics. So here are the three broad categories are divided in the methods of bacterial characterization. The first one is molecular biological methods, and the second one is biochemical methods, and the third one is microbiological methods. So let's briefly discuss about these three methods. The molecular biological methods including single or multiplex PCR, which stand for polymerase chain reaction, and real-time PCR, nested PCR, reverse transcription PCR, post-spiritual electrophoresis, multilocus sequence typing, plasmid profile analysis, and random amplified polymorphism, deoxyribonucleic acid, which is called also RAPT. The biochemical methods depends upon the differences in protein and fat metabolism, carbohydrate metabolism, enzyme production, compound utilization ability. So there are some most common biochemical methods depending upon enzyme production. The first one is catalase test. In this test, many aerobic bacteria produce an enzyme known as catalase that detoxifies hydrogen peroxide, and the positivity of test is determined by gas O2 formation in form of bubbles. Then in oxidase test, bacteria which function on aerobic respiration often possess cytochrome C and cytochrome C oxidase. The test uses oxidase reagent applied filtered paper, timely color change of which indicates the positivity of result. And in indole test, the bacteria possessing an enzyme tryptophanase can hydrolyze with amino acid tryptophan to form indole, and the ammonia and pyruvic acid. The timely color change during the test determines the positivity of the result. Then microbiological methods of bacterial characterization include the gram staining methods and spore staining method. Gram staining is a common technique used to differentiate two large groups of bacteria based on their different cell wall constituents. The gram stain procedure distinguishes between gram positive and gram negative groups by coloring the cells red or violet. And gram iodine is used in gram staining procedure to differentiate gram positive and gram negative organisms. The grams iodine act as a moderant that causes the crystal violet to penetrate and adhere to the gram positive organism. Then the endospore staining is a technique used in bacteriology to identify the presence of endospores in a bacterial sample. Within bacteria, endospores are protective structures used to survive extreme conditions, including high temperatures, making them highly resistant to chemicals. And this staining is the type of staining to recognize the presence spore in bacterial vegetative cells, and the bacterial endospores need a staining which can penetrate wall thickness of spore bacteria.